Good evening. My name is Dr. Shakti, and we are going to be covering a very important topic in respiratory system, that is regulation of respiration. So you can take a look at these various questions to get an idea as to what sort of questions usually come from this. You can, of course, see a long essay where you have to talk about neural regulation of respiration in total. So if that sort of a question comes, then we have to divide the topic into how regulation of respiration is happening at a neural level, it is happening at a chemical level. And both of these being involuntary in nature, but the third point being some form of a voluntary control also being involved. So under these three headings, we have to discuss the entire set. Then we have shorter questions. You can see, again, specifically, they can ask you about central chemoreceptors, then peripheral chemoreceptors. So each of that separately can be asked as a question. One specific extra thing that you need to learn is about Herring-Brewer reflex. And something called hypoxic drive. What exactly is hypoxic drive? So we'll come to learn all that when you talk about your chemoreceptors. Then the respiratory centers located in the brainstem. So this is something that comes under neural control, and explain how blood pH influences respiration. That is also your chemoreceptors. So as you can see here, these are the various headings under which we are going to try and discuss this topic. Okay. So as I was talking about it earlier, we have a involuntary control, which includes your neural respiration regulation, and we have your chemical. And then we have voluntary control. These are the headings under which you have to discuss this. So we'll start off with your neural control. We'll just start off with a diagram of your brainstem, not including the midbrain over here. We have your pons, your medulla. I'm just dividing it into two for ease of understanding. And below that, we will draw the spinal cord. Okay, so this is the system that we are dealing with right now. So in your medulla, most of your respiratory centers are either there in your medulla or certain parts in your pons. In your medulla, right in the center, we have an area which is nucleus, fructus, solitarius. This is where all the sensory information is coming and getting interpreted before it is going to various parts, especially in the brainstem, for various types of reflexes. Just lateral to it, we have a group of neurons over here. On either side. So there are a group of neurons on either side, just lateral to your nucleus fructus solitarius, and these are what are called your dorsal respiratory group. We call them dorsal respiratory group of neurons. These group of neurons, what they do, they provide a stimulus which is taken to the other side, comes down, taken through the white matter, and eventually it will go to the ventral horn, and from there it is going to be taken to a muscle. Which muscle? The primary inspiratory muscle is your diaphragm. This is what your dorsal respiratory group is doing. And in terms of diaphragm, we are talking about your eupnic breathing right now. That is your tidal respiration. So what does your dorsal respiratory group do? Dorsal respiratory group results in activation of the diaphragm to result in eupnic inspiration. We have already learned previously that expiration is a passive process. So just the stop of the signal will result in a recoil, and expiration passively can happen. So for a eupnic setting, for tidal respiration, dorsal respiratory group is going to result in inspiration. Once it stops acting, it is going to result in your expiration just by itself. This is your dorsal respiratory group. Now, apart from the dorsal respiratory group, even more laterally if we go, we have a ventral respiratory group. Now, this ventral respiratory group has first a complex over here, which is, having two parts. On top, there is a Botzinger complex, and we have the pre-Botzinger complex. So we have the Botzinger complex, pre-Botzinger complex. Beneath that, we have the nucleus ambiguous. Next to the nucleus ambiguous will be a para. So a nucleus para ambiguous. And below that, you will have a nucleus retro ambiguous. Okay, so what do we have so far? Botzinger complex, a pre-Botzinger complex, nucleus ambiguous, nucleus para, next to it a nucleus para ambiguous, a nucleus retro ambiguous. This whole thing together will form what we call the ventral respiratory group. Now, how does the ventral respiratory group act? This is where if you need any sort of overdrive mechanism, where you need to take in a deeper inspiration or exhale more forcefully, then that is when your ventral respiratory group is going to come into play. So you can see here, once you draw this, whatever is at the edges, the Botzinger complex, which is over here, and the nucleus retro ambiguous. These are the ones which result in information which is going to be carried and will result in your expiration. So this expiration again will go here like this and come out, and it will go to your rectus abdominis. It will go to your transverse abdominis. It will go to your oblique muscles, internal and external oblique, and we have the internal intercostal. All of these are expiratory muscles. What happens is finally your Botzinger complex is going to, result in stimuli, and as you need more and more recruitment of more and more for more forceful expiration, it will start recruiting neurons from the nucleus retroambiguous as well. On the other hand, for inspiration, if you want more overdrive inspiration, not just with your dorsal respiratory group, if you want more of that, then your pre-Botzinger complex is going to come, your nucleus paraambiguous is going to come. So what do? They will again go and act. In a similar way, where do they act? They go and act on your external intercostal muscle, all your accessory muscles of inspiration. What are they? Your trapezius, sternocleidomastoid, scalenus, your ala nasi. Okay, so all of these accessory muscles of inspiration will be recruited when now the signal goes from the pre-Botzinger and if needed more, your nucleus parambiguous. And there is an inverse relationship where, when Botzinger complex is active, it will inhibit the pre-Botzinger. When pre-Botzinger is active, it will inhibit the Botzinger complex. So in that regard, while inspiration is happening, expiration is inhibited. While expiration is happening, inhibition of your inspiration is happening. So there is that sort of a inverse relationship over there. So what does your nucleus ambiguous do? That is also involved in inspiration. That is more towards your pharyngeal laryngeal muscles. Okay. So that is your dorsal respiratory group. We have your ventral respiratory group, and how the interplay is happening. Dorsal respiratory group is for your euphonic inspiration for the diaphragm. And then if you need overdrive excess inspiration, your pre-Botzinger complex, nucleus parambiguous will act, and nucleus ambiguous will also act on your pharyngeal laryngeal muscles. And then if you need expiration, just dorsal respiratory group stops functioning, means expiration will take place. That is tidal expiration. If you want more of an expiration, then your Botzinger complex, nucleus retroambiguous is going to come into the play. Okay, so this is what you have to understand about your neural respiration. But we are not done with this. This is where you have to understand a terminology called a RAM signal. So ultimately, what you see is in Botzinger, pre-Botzinger, dorsal respiratory group, all of these areas, you start seeing some RAM signals like this. It could be, when you look at it, it'll be like this. Okay, these are what are called RAM signals. And these RAM signals are what gives rise to the impulses for the activation of these muscles. Now, how does the RAM signal arise? Actually, there is a model which is widely accepted, which is called emergent property model. Different theories are there, but the most widely accepted model is this. What it tells is, different parts of your medulla, there are different kinds of receptors. Okay. So these kind of neurons from different parts will now come and act. And when they act together, that is how your respiratory signals are rising. And based on how much of the activity is happening, how long do you want it to go on, based on that, you can fine-tune the signal. So when a ramping is happening, this part, that is when your inspiration like that is happening. Okay, longer the ramping, more the period of inspiration. So this is a group of neurons. This is the early inspiratory, augmenting, late inspiratory, expiratory, decrementing. Okay, and we have late expiratory, pre-inspiratory. So like that, there are different neurons which are involved, which creates the RAM signal. Now, the peculiarity of this RAM signal is this is where you have your control from your pons. So there are two centers in the pons. One which is called the apneustic center, and another one which is called the pneumotaxic center. What does the apneustic center do? Apneustic center makes this ramping keep going on. So if ramping keeps going on, just imagine what will happen. The person's taking a breath. And he's keeping on taking a breath. At some point, you cannot take a breath anymore, but still, there is no breath. He's stuck in that position. So that results in a condition called apnea, cessation of breathing. Although inspiratory drive is increased, it'll get stuck over there. That's why it's called the apneutic center. That is what apneutic center does. Pneumotaxic on the center, on the other hand, will inhibit this apneutic center. So in a way, it can regulate the control as to how long the inspiratory drive should happen. So it can go as short as 0.5 seconds. It can go as long as 5 seconds. So this is how much variability your pneumotaxic center is affording you. Okay, so a pneumotaxic center can cut short and inhibit the apneutic center to make it stop much more earlier at this point, or it can let it be and make sure that it will last long. So based on that, the inspiratory drive is going to change. So now you understand the entirety of your neural regulation. Now that neural regulation is understood, we'll go to the chemical regulation. I'm going to draw a system over here. Let's just take your lungs here. I'll put it in a small box like this. And I'll try to encase it outside. This will be your rib cage, your mechanics, the entire mechanics. And what will be here? Your muscles in between. So these are all the muscles we were talking about earlier. All these muscles. Now they regulate how much of ventilation can happen according to the neural regulation. Now, what does this essentially do? The whole point of your respiratory system is to make sure that your oxygenated blood is getting oxygenated and goes into a cell. You need to give oxygenated blood. If you look over here, you will see that generally your partial pressure of oxygen will be higher, partial pressure of carbon dioxide will be lower here. By the time it gives to the cell and comes back, PCO2 will be higher and PO2 will be decreasing. So this is the whole function of your respiratory system. Now, this is where you need to keep some sort of a receptor in the periphery. Okay, mind you, again, we're talking about chemoreceptors, chemical regulation of respiration. First type of receptor is a peripheral receptor. Central means it will be somewhere in the brain. But in this cycle, somewhere in the outside, we now need to understand, okay, is the PO2 that was given enough? Did I oxygenate it well enough? If not, I have to inspire more air. Okay, or instead, if PCO2 is higher, you need to realize, okay, I need to respire faster to wash it out. So like that, you need some sort of peripheral feedback. So that feedback is through a small body like this. Consider a small body which is arching out. What will happen? The blood which is flowing like this and reaching here. A part of that blood will go through this and will cycle and come back. And although this is actually a very small part, it is receiving one of the highest blood supply per unit area if you look at it in the body. So what does it detect? It helps you understand about how much your PO2 is there, how much PCO2 is there, and how much is your pH. These are three things it majorly detects. Now, these cells, these bodies that we call, can be of two types. They're in two locations. We have the carotid body and we have the aortic body. We have the carotid body, we have aortic body. So these bodies are chemoreceptors. Instead, if you have carotid sinus, that is a baroreceptor. And what type of chemoreceptor? A peripheral chemoreceptor. Now let's be more specific, go into it. So when you reach this body, you see that at the edges, there are a particular group of cells which are present. So these are some cells you will see here. These cells are called glomus cells. And this, which I have drawn in particular, is what is called type 1 glomus. Apart from that, there is also another type of glomus cell. This is what you call a type 2 type of glomus. You have type 1, type 2 glomus. Type 2 glomus cells are sustentacular. They just support it. While you have the major type 1 cells. Now they detect, as I was mentioning, your PO2, PCO2, and pH. Now from here, we have innervations which are arising. Now terminals, which carry certain information back. If it is a carotid body, your 9th cranial nerve. If it is an aortic body, your 10th cranial. Okay, if it is a carotid body, it is a 9th cranial nerve. If it is an aortic body, it's your 10th cranial nerve. Now, on these glomuses, we have a particular type of channel, which is a potassium channel. What does potassium channel do? It always represents potassium efflux. Now, this channel, one thing, it is oxygen sensitive. So say oxygen is less, PO2 is less. The channel is no longer active, resulting in more accumulation of positively charged ions inside. These positively charged ions inside will make the threshold go up in order to open up a calcium channel, because of which calcium will now come in. As a result of that influx of calcium, there are vesicles containing certain transmitters, which are now ejected outside. And that will eventually go to the other side and stimulate the nerve terminal. And the signal keeps getting carried up, and by no surprise, it goes and reaches this area we were talking about, which was the nucleus tractus solitarius. So all the signals which are coming up will come over here. And then it decides, okay, the response was that PO2 is less, we need to increase the inspiration. And then it tells the DRG, if we need more, it will now go and tell your pre-birthing nucleus paraambiguous, nucleus ambiguous. So this is how your peripheral chemoreceptors work. Now, apart from that, what about your PCO2 pH? In a similar way, like how we were talking about oxygen sensitive, if CO2 is high, CO2 will come inside, that will get converted to H2CO3 and will give H plus eventually. Or H plus is there outside, H plus comes in. Eventually, this can also inhibit your potassium channel, resulting in the same sort of events that we are talking about. Now, there is a little bit of controversy regarding what is the neurotransmitter which is involved there. Even some of the standard textbook is giving one, while another one is giving another answer. So majorly, if you have to talk about it, purinergic transmitters like your ATP will be more important one. Gannon, however, mentions about dopamine acting on a D2 receptor and all, but most studies where dopamine is seen is usually in certain animal models, especially cats and all. Okay, in human beings, you see that ATP is more important. Okay, you can write them both in case somebody asks, needs one answer. The better answer would be ATP. Otherwise, dopamine is also the second option. Okay, but it makes more sense to talk about ATP. That is your peripheral chemoreceptors. So let's just take a small break to take a look at some of the questions. So the respiratory center located in the pons, that smoothens respiratory rhythm. Smoothens as in some sort of control it is trying to do is what they are implying. Especially since they're talking about pons. So what are the things that can regulate the rate? That is your pneumotaxic center. As low as 0.5 seconds to as long as 5 seconds. Which respiratory center stimulates forced expiration? Now, this is a very vague thing again. It's a ventral respiratory group. But which part in ventral respiratory group? Again, this question answer options can come very different. So be, knowledgeable about that. You have to specifically say that in ventral respiratory group, your word sinker with the retro and ambiguous will be important if a forced expiration is required. Dorsal respiratory group of neurons is primarily involved in, so you know that it is for your normal respiration. That too, inspiration. Okay, out of the option, the best option will be you're generating your inspiratory rhythm. Okay, so I'll just leave it there. We'll go back to your chemoreceptors. So this is one type of chemoreceptors, which is called your peripheral chemoreceptors. Now, apart from the peripheral chemoreceptors, we also have a central chemoreceptor. So for that, we'll have to go back to this area. I'll just rub these off. I'll just draw a blood-brain barrier over here. So as you should be knowing, everything that is coming through blood should not directly go and reach your brain. There is some sort of a blood-brain barrier which is there in between. Say, for example, your H+. That by itself cannot cross this blood-brain barrier. More soluble elements, not ionic elements, soluble elements like your CO2, oxygen, they can in turn go inside. Now the thing is, your chemoreceptor area is there in the medulla. In the medulla, we have 3 areas ventrolaterally here, like this. 1, 2, 3. So ventrolaterally, we have 3 areas. Okay, the name you don't have to remember. I'm just putting it down in case somebody needs. This area is called Mitchell. This area is called Schlafke. And this area is called Loschke. Okay, again, not required of you to remember it, but if you need it, you can use it. So there are 3 separate areas. These are their ventrolateral part of the medulla. Now, they are sensitive to H+. But as we were talking about, H+ cannot come inside. Instead, what happens? The CO2 comes inside. So this CO2 which is there in the blood, so CO2 in the blood will indirectly be detected by converting it into CO2 with water will form H2CO3, which will split into H plus HCO3 minus. And this H plus will go and stimulate these centers. This is your central chemoreceptors. Central chemoreceptor. Again, questions can come which will try to confuse you. Again, H plus is the one which is directly stimulating the central chemoreceptor, but it is indirectly taking into account the PCO2 of the blood. This is where you have to understand another terminology, which is hypoxic drive. This is called hypoxic drive. So generally, if you look at it, your central chemoreceptors are around 7 times more powerful than your peripheral chemoreceptors. Why? What is the whole point of peripheral chemoreceptors? They are 5 times more rapid. They're faster. So fast detecting stuff, that is peripheral chemoreceptor. But much more powerful is your central chemoreceptors. So which would be the major signal in our body to determine regulation of respiration? That would be your PCO2. Generally, a PCO2 is the major role when you talk about central chemoreceptors. This is where another terminology called hypoxic drive is coming into play. Especially when your PO2, it starts falling and falls less than 60. PO2 falls less than 60. Then that can in turn result in a respiratory inspiratory drive, which is called a hypoxic drive. There, they're not looking at PCO2 anymore. Your PO2 level falling by itself will stimulate this. That is what's called hypoxic drive. Because there was one question earlier. Okay, so regularly, the PCO2 is a major role when it comes to regulation of respiration, more powerful. But if the hypoxia is there and PO2 is falling less than 60, then that can impart an inspiratory drive, overpowering your PCO2 values. That is your hypoxic drive. So now you understand about, if you take a look at these diagrams, we talked about neural regulation, we talked about your peripheral chemoreceptors for chemical regulation and the central chemoreceptors for, again, chemical regulation. Now, we'll go on to the remaining questions. So, increased ventilation in response to metabolic acidosis is mediated by. So, metabolic acidosis is there. So, one of the things about metabolic acidosis is your H plus is increasing. If H plus is increasing in the body, that is going to stimulate your peripheral chemoreceptors. H plus in the blood cannot stimulate your central chemoreceptors. But when increased H plus is there, something you learn in your renal system, we have a buffer system in our body. One of the buffer systems is a bicarbonate system, most important buffer system. So what happens is when H plus is increasing and a metabolic acidosis is happening, the bicarbonates which are there in the system will try to combine with that. And so your HCO3 minus will come to reduce the amount of freely available H plus which are there, just forming H2CO3, which will split into CO2 and water. And that CO2 again results in increased PCO2. That can again stimulate your central chemoreceptors. So when there is an increase in ventilation in metabolic acidosis, there is both the involvement of your central as well as peripheral chemoreceptors. Peripheral chemoreceptors are located in, so we were talking about it, the carotid and the aortic bodies. Central chemoreceptors are more sensitive 2. Okay. So again, if you look at this question, one thing is when you look at this, you might tend to go for this answer. Okay, because PCO2 is one of the things. So this is what I was telling you earlier, how they can try to form questions in order to confuse you. But remember, the question here that is asked is central chemoreceptor. That central chemoreceptor, what is it sensitive to? The H plus. The H plus, which is there after crossing the blood-brain barrier. So that will be the best answer here. Okay, instead of that, if they're talking what in the blood is gonna affect the central chemoreceptor, then the answer would be this. Now, this is another thing, hypoxic drive. So hypoxic drive, already I mentioned to you, so what is happening when PO2 is falling below 60 millimeters of mercury, then that stimulates your hypoxic drive. Next, we have to talk about something else, certain sort of reflexes that can happen. One is what is called Herring-Brewer reflex. Before that, we have the involvement of your vagal system. Over here, when we were talking about your neural regulation of respiration, something I have to add over here. That is the vagus nerve. Your vagus nerve will result in a similar sort of activity as a pneumotaxic center. As long as either pneumotaxic center or vagus, one of this is still there, still the apneustic center can be inhibited and kept. If both of them are gone, then there is no control on your apneustic center and it will result in apneusis. So both vagus and pneumotaxic center have a similar sort of role, whatever is coming from the vagus. Now, that is what we are talking about now. Signals which are going to come through this vagus. For that, we have first one thing called a slow adapting. Slow adapting kind of receptors are there. Then there is something called a rapidly adapting. So slow adapting receptors, rapid adapting receptors. These are usually both myelinated. And a third variety, which is C fiber, which is unmyelinated. Three type of channels are going to be there. And a common example of this is through something called a J receptor. We have a slowly adapting kind of a receptor. We have a certain rapid adapting receptor and a J receptor. What do these three things do? That is what we're going to understand. So if you look at your tracheobronchial tree, it's going into the lungs and it's for the splitting. Remember the first class, how we were talking about the tracheobronchial tree. And finally, your alveoli. Now, stretched all over these areas, your bronchus, your bronchioles, terminal bronchioles, respiratory bronchioles, even your alveolar duct and parts of the alveoli, all throughout the tracheobronchial tree, we have certain types of receptors. These receptors are the slow adapting receptors. What do they detect? They detect stretch. So when there is sudden inflation, which is a sort of a hyperinflation. So sudden increase in more than, usually we give a value like more than 1.5 liter. But again, there are different studies which are showing, different values. But for your level, you can remember this value, 1.5 liters. A sudden inflation of more than 1.5 liters will result in stretch through the slowly adapting receptors. Through the myelinated nerve fibers, will carry it, will go through the vagus, and will come back down. This will go up and then inhibit your inspiration. So when sudden hyperinflation is there, if this keeps happening, that will result in damage to the tissue. So to prevent that, this reflex will now go and inhibit that inspiratory drive. This is especially seen in preterm and in infants. And usually, by the end of 1 year, this kind of reduces. In adults, you don't see it so much. It's still there, but you don't see the effect of this as much as you see in your preterm and infants. What is the stimulus? Any sort of hyperinflation or sudden occlusion that is happening. Okay, so this sort of stretch which is happening. So this is your slow adapting receptors, and this is what we call Hering-Brewer reflex. Now, the opposite of it, a sudden deflation. A sudden deflation is problematic because remember when we talked about compliance curve, at lower end of the compliance, it is more difficult. The pressure that is needed to maintain that is more difficult. Surface tension increases, and there is higher probability of collapse of the alveoli. And once collapsed, it is very difficult to open up again. So it is essential that when sudden deflation is happening, you do not reach that level. It needs to be stopped before that. So a sudden deflation will result in a inspiratory drive. That is reverse of Hering-Brewer. Okay, so that is like two things: sudden deflation, sudden inflation, and how Hering-Brewer reflex is happening over here. Now, the second type is a rapidly adapting. Now, these are there not in the tracheobronchial tree per se, but in the epithelia which is there. So here again, I think I forgot to mention it, in the tracheobronchial tree specifically where near the smooth muscles. Okay, I think I had forgotten to mention it. Now, these rapidly adapting receptors, they will be seen in the epithelial layers. Over here, the epithelial layers which are here and here. So all of these epithelial layers, they have the rapidly adapting receptors. What do they detect? Any sort of irritant. So these sort of irritants, if it's there in the upper respiratory tract, that can result in something like a sneeze. If it's coming in the lower respiratory tract, that can result in a cough reflex. So any sort of irritants which are coming result in some sort of sneeze or a cough. That is through these rapidly adapting receptors. And then we have the J receptors. Now, over here, between them, there is the interstitial fluid here. So over here, we have the interstitial fluid with collagen, collagen, all that is here. So over there, we have a particular type of receptor. That receptor is which is called your J receptors. The J receptor was actually discovered by an Indian scientist, A.S. Paintal. So this, what does it detect? Accumulation of fluid. So when there is excess accumulation of fluid, these J receptors detect that. So any sort of pulmonary congestion which is happening. Why pulmonary congestion? When the blood is not flowing properly through the pulmonary circulation, then the hydrostatic pressure over there increases. More hydrostatic pressure means more of filtration and fluid will accumulate in the interstitial fluid. Or sometimes even some sort of a heavy exercise. This can be detected by these J receptors. So again, the effect is a vagal sort of thing. Immediately, there will be a apnea. First impulse is that apnea. What does this vagus do? It results in a bradycardia. This is the first impulse that happens. Now, because apnea happened for a long time, there will be buildup of PCO2. That buildup of PCO2 will now be detected by this central chemoreceptors to trigger a rapid inspiration. Apnea followed by a hyperventilation. So this is what happens as a result of activation of J receptors. And even for that matter, even your rapidly adaptive receptors. Now, take a look at this question. Heron-Brewer reflex is mediated by stretch receptors located in. So this is what I was mentioning. So you have to specifically understand not only bronchus or the bronchioles, it could be, even the alveolar duct and terminal or respiratory bronchiole, all of these areas, smooth muscles have these stretch receptors. Which of the following is characteristic of the Heron-Brewer reflex? So what does it do? It basically prevents overinflation of the lungs. Okay, so if you remember the questions, we started off with all the neural regulation, the role of central chemoreceptors, peripheral chemoreceptors, what is Heron-Brewer reflex. We talked about hypoxic drive. Then we talked about the neural regulation under which the respiratory center is located in the brainstem. How does blood pH influence respiration? You can talk about it with regard to your peripheral chemoreceptors as well as your central chemoreceptors. Okay. So does anybody have a doubt? Please let me know if you have any doubts. So we're just going to talk about Auricle. So Auricle has foundational classes which are there, and all pre-recorded sessions are over there. Live sessions are there where you can actually use it to interact with the teacher, get the feel of being in a class, and ask doubts. That is the main thing about live sessions. Express exam module is there. So, right before the exam, if you want to understand like what are the important topics, what do I need to stress upon, what are the usual questions that are asked, this is the go-to one where within 20 hours, the entire physiology is done. There are one-to-one doubt clearing sessions, and there are hard copy notes which are made available. And there are question banks as well. And these are for various difficulties. Even pre-recorded practical sessions are there. And these are the current subscription plans which are there. And you can use my code, Dr. Shakthinag 20 for discount on the subscription. So again, thank you for watching. You can contact Team Auricle through any of these social media handles if you have any queries.